plan for today was to do a workshop after the talk, which was going to be fun and hands-on. Unfortunately, we can't do that, so I'm going to have to tell you about the workshop, which might sound a bit strange, but actually it's going to be really helpful for me to explain things to you, and then you can give me some feedback at the end. So I work in the molecular lab at the Natural History Museum, and odd things are happening to my screen. Hang on, let me just shrink that. Excuse me a second. Okay, so here at the Natural History Museum, we have a DNA sequencing lab, and you can come and see us. If you go into the galleries, you can actually look at us through the window, which is a little bit odd, but it's quite fun once you get used to it. And we do DNA sequencing. We provide a sequencing service. We have different methods of sequencing. We do Sanger and Illumina and MinION sequencing, but they all basically do the same thing. They're just different methods of looking at DNA. We love our outreach. Here we are doing some Nature Live presentations. So this is something we do in the galleries when we tell people about the sequencing facility. Here we are talking about environmental DNA to the public. We also do schools workshops. This is a pop-up Meet the Scientists that we did in the galleries back in January. And this is European Researchers' Night. This is always really good fun. We do DNA extraction from fruit. So we get members of the public to come in and extract DNA themselves, and they can physically see the DNA once it's out of the fruit, and they can take it home with them. So this is really good fun. Children really enjoy this. It's something they can do, and because they can see it, it's much easier to explain what's happening because you can talk to them whilst they're doing it, and they can see and they can feel the actual process of DNA extraction. Unfortunately, when you're trying to talk about DNA sequencing, there's not really a huge amount that you can do and see for sequencing. So we tend to lose kids. Here we are talking to some adults. Adults are always really interested, and we get lots of questions, and it's really good fun. But you know, children, it's a lot harder. We came up with this game. My colleagues Darren and Sarah made this great cardboard DNA sequencer and made a little game to guess the DNA sequence. And it was fun, but we're always looking on ways that we can improve our public engagement. And that's when I discovered the Brickopore from Bloxford. So it was designed by some people at the Earlham Institute for basically this purpose that we wanted. And it's a model of a DNA sequencer that's built entirely out of Lego, and it's robotic, and it sequences Lego DNA. So you build DNA out of Lego bricks, and then you run it on this sequencer. And it's just a really good way of explaining how actual DNA sequencing works. Obviously, you can't buy it as a kit because it's only just been invented. You have to buy this Lego Mindstorms Kit, which is a couple of £100 because it's got robotic sensors and all kinds of other things in it. And then you have to buy extra little bricks, and then you put it together. All the instructions are on the Bloxford website, tells you how to make it, and then there's software that you can download and you can put on your laptop. It just runs off a standard PC or Mac. So Earlham have used it quite a few times. Here's some images from their website of different public engagement activities they've done. You can see it was a great success, and I've stolen quite a lot of their ideas to make our public engagement activity. So I'd made this activity, and we were going to run it at the Exhibition Road Festival, which is this photo here, but obviously it was cancelled this year. So that means that we haven't had a chance to try it out. I took it home and tried it on my kids and tweaked it a little bit. But actually, I'm going to run through it with you now to see what you think. And if you've got any comments or questions, that would be really helpful in how we move this thing forward and actually do our first workshop, hopefully in the new year. So how are we going to explain environmental DNA to children in the galleries using Lego? So our first step is to explain that DNA is a bit like an instruction manual. It tells you how to assemble a living thing. Now, every living thing has DNA, from amoebas to amphibians, from polar bears to potatoes, from boys to beetles. Okay, and every time a living thing molts or emerges or spawns, scratches, drools, sneezes — I like to think that bubble is the new sneezing — does a poo, or dies, some of its DNA comes out of it and is released into the environment, which means that in the environment there is DNA. So it's in our soil, it's in the sea, it's in ponds and lakes, rivers and streams, and it's even in the air. And if we can capture this DNA, we can get a good picture of what animals are in our environment. So for soil, you have to extract the DNA from the soil using kits like these. It's a chemical or filter-based extraction to wash the soil away from the DNA. Air is a little bit easier in a way because we don't have to wash anything out. We just use this this machine here. It's called a bobcat, and it's got lots of fans in it, and it basically sucks air through a filter. You have to suck a lot of air through a filter to get a reasonable amount of DNA, like a lot, but it does work. And here's water. I believe these are Lindel's hands actually, collecting some water from a stream and then using syringes to force it through a filter, and then you get the DNA off the filter. So since every living thing has DNA and every living thing is unique, the DNA of every living thing is unique. So if we can extract it from the environment and read it, we can then identify which animal it came from. So we can get a picture of what animals are living in an environment. Now, when you think about it, DNA is a little bit like Lego anyway, in that it's made up of different blocks. So the Lego DNA that we use on the Brickopore is made up of these 4 different colors, and we use a different color to represent each of the nucleotides that you would get in a DNA strand. Now, before you do DNA sequencing, you can't just put DNA straight onto a sequencer. You have to manipulate it in a certain way so that the machine can actually get hold of it, can actually read it. So for the Brickopore, it's the same thing. You have to make sure that your Lego DNA is exactly 22 bases long and that it has 3 white bricks on the end as an adapter. So let's hope this works. Yes. So you put your DNA Lego bricks onto the machine. You press sequence, and it pulls the DNA through the machine in the same way that, say, a nanopore sequencer would pull a strand of DNA through a pore in a membrane. This pulls it through the robot. And then the software on your laptop generates this thing called squiggle data, which is based on a nanopore sequencing. So that's exactly what you'd see if you were using a nanopore sequencer anyway. And then the squiggle data is converted into base calls, which is how you get the DNA sequence, which appears at the bottom in bright colors. Yep, so there's our squiggle data. Once you've got your sequence, you then hit blast at the top, and that will take you to the NCBI website. This is a GenBank database, and it cuts and pastes your sequences into the GenBank database. It does a search, and then it looks for your sequence. So this is the kind of thing that BLAST gives you. You can see that that particular fake DNA strand that we've made was a match to Anopheles stevensii, and so that's the one that we would tell the kids, you know, your DNA is from this particular animal. Now, unfortunately for us, the NCBI gives everything in taxonomic names. So for children, that's a little bit strange. So for this step, you then have to cut and paste that word into Google. But I found practicing this with my own children that actually that wasn't a problem because they're quite used to cutting and pasting things into Google anyway. And when you do that, Google often brings up this nice little picture. So for my youngest son who can't read very well, that was really great. He could put this in and go, oh, I've made the DNA of a mosquito, or I've made the DNA of a lion, and he could see the picture, and he really enjoyed it actually. So this is just a video of me practicing this activity with my kids. So my kids don't actually like science, which is a real shame, and they don't really understand what I do. But taking this home and practicing with them was really good fun. They really enjoyed it. It was Lego, which is something that they're used to, so they weren't afraid of using it. They're used to using Google and laptops, so again, it was something they were used to. They weren't afraid or embarrassed, and because it's something tactile and it's something they can do and something they can actually see, I found it much easier to explain how DNA sequencing works. And they were actually really engaged with it, and they carried on doing it for ages. So I'm really, really hopeful that this particular engagement activity is going to be successful and that we're going to be able to use it a lot in the future. But if you've got any suggestions on how I can improve it, if there's any steps that didn't make any sense, then I'd love to hear from you in the chat window. That would be great, or you could ask some questions. And also, since everybody loves DNA, it's not DNA, everybody loves Lego, I'm thinking that actually this is going to be great for adults when we do our evening activities. And that's a photo of, of people in the galleries having fun, and our actual Lego sequencer with all our little minifigs. So, and that's the end of my talk. If anyone has any comments or questions, that would be great.
Hi, Andy. Thanks very much for that. That was brilliant. And I think you should leave that slide up, because if you notice, all these little Lego minifigure lab technicians, they're actually wearing these little blue nitrile gloves that you would wear if you were really in the lab. So I just thought that was really sweet when I noticed that.
They are all modelled on the lab team. Actually, we made them specially out of parts so that they looked like members of the lab team.
So
yeah.
Where are you in this picture, Andy?
I'm bottom right. My hair's grown since then.
Oh, this one.
I can't see. I'm still sharing my screen. So.
Are you looking a bit boyish in this picture, Andy?
Yeah, I've got short brown hair, and I'm on the right-hand corner. I did have very short hair when I made it.
Okay, so yeah, I think it's a real shame that we weren't able to do this as a proper workshop, because I think people would have had fun making their Lego molecules and actually doing the sequencing. And I think the process of actually doing it does answer some of the questions that people, because it is quite a complicated process, isn't it? And it does. In this, your Brickopore sequencer, what is it a sort of facsimile of? Is it, because we've heard a few different names of different kinds of sequencers rolled off. What's it closest to?
So I guess it's a cross between a Nanopore sequencer and a Sanger sequencer. So in Nanopore sequencing, the DNA is split into two halves, and one half is threaded through pores inside a membrane. And as it's pulled through the pores, the membrane detects exactly which base is going through at that time. And then the data that you get with the squiggles, that is a facsimile of what you would get from a nanopore, and then the base calls are based on that as well. And since this is called a brickopore, I think that's what they were going for, the guys who invented this. But obviously, the way it actually works is with a little color sensor, and it detects the colors of the bricks that pass through. And that's how Sanger sequencing works, and in a way also Illumina sequencing, by you use chemical processes to attach colors to your DNA, and then you read the colors. So in that sense, it's similar to Sanger, but the squiggle data and the movement of the DNA through is based more on nanopore sequencing.
Okay, so these different styles of sequencing your DNA, are we using different styles of sequencing for different aspects of the Darwin Tree of Life project? Is that something that you have?
I believe most of it's going on the PacBio, which we do have in the basement. I didn't mention that one because we're not offering that as a service. We just bought it specifically for the Darwin Tree of Life. I think that's probably one more for Lyndall and Mara, but I think that is all going on the PacBio SQL system. So that's something called smart sequencing. You'd have to ask Mara about that one.
Okay. Well, we can, yeah, don't worry, we can answer more questions after we finish the session. Okay, so there are a few questions coming up in the chat, so I will read them out. We have a question from Linda Pryke. Andy, maybe a challenge as to who can DNA the largest / fastest / tiniest cool animal. I think this is for the children, though possibly for the adults as well. I mean, I am yes. Quite sad that I'm not now in the Angela Marmot with you, Andy, playing with DNA. Yes, I was looking forward to that. That was going to be the highlight of my day.
My kids did get really excited. Actually, they were doing this for ages, just seeing what they could make. And I think the highlight was the they've got so many animal toys. I mean, some of the photographs I showed you were their toys that I used as an explanation. And they managed to make a langur, which is a type of primate, which is their current obsession. So they were so over the moon that they'd made a langur. And
so it got quite exotic then.
Yes.
Okay. A question from Barbara and John. Could you also explain something about the problems with sequencing, e.g. cleaning up the sequence? Now that I've read that out, that's probably quite technical. Is there a summary of the stages that need to happen?
Yeah, I think that would be something we talk to about adults rather than kids, because it's quite complicated. Cleaning up the sequence. If you're doing Sanger sequencing, you use something called PCR beforehand, which should help clean up your sequence and just give you the one thing, but it is quite prone to contamination, so you do sometimes get these extra peaks from contamination, and that's very difficult to clean out. If you're doing NGS, so Illumina sequencing, PacBio sequencing, that kind of thing, you have to clean that out later in your data processing. So each individual sequence is clean, but you might have 100 sequences which are supposedly the same thing, but they will vary slightly. So you have to use various bioinformatics tools to clean out your data, to clean up the sequences. I hope that answers the question.
Yes, I think it's easy to get confused because there are these different sequencing technologies, and depending on which one you use, it has different advantages and different disadvantages and different limitations. So you can read that someone did some sequencing and there was this particular limitation, and actually that only applies to one particular style of sequencing. So it is quite a complex thing, isn't it?
Yes, exactly. I mean, we currently use 3 different methods in our labs, depending on what question you're asking. And yes, there is this extra one, which the Darwin Tree of Life is using as well. So yeah, there are currently 4 methods. We're also hopefully in the process of buying another method, and then there are at least 2 or 3 others out there. There are so many different ways of doing this. It all gives you a sequence in the end, but they all have their different advantages and disadvantages.
And there are also complications in the kinds of samples that people are using as well, aren't they? Because some sequencing technologies are applicable if you're sampling a particular organism, and then other methods will be used if you're sampling this environmental DNA, which is obviously going to be a mixed bag right from the beginning. You haven't separated out your different organisms, so it's a different beginning, and you're asking different questions, and you're going to get different answers. So it's
for environmental DNA, you would have to use a next generation sequencing technique. Sanger wouldn't work, but we've used Nanopore, Illumina, and the PacBio SQL system would all work quite well for this.
There's a question here about the differences in scale. I think this is referring to the fact that your Brickopore sequencer can only do quite a short DNA molecule. So have you had any thoughts about how you're going to explain to the children the huge differences in scale between what you can do with your Lego and how big an actual genome will be?
Yeah, so I had a chat with this about my son. So yeah, you can only do 22 nucleotides on the Brickopore, which is far too short practically for anything. So if you saw on the slide where we had the Anopheles one as the top hit, at the bottom there was a breakdown of the different hits, and it was also pulling up fish and bacteria. So 22 is just far too short. But that's how the Brickopore works. You'd have to buy an awful lot more Lego to make it huge. You'd have to make it the size of the table to have anything of any meaningful length. But yeah, if you were doing, say, Illumina sequencing, you'd have to shear it. As Mara explained earlier, you do have to shear the DNA to be the optimum length, depending on your particular platform. So Nanopore sequencing can handle very long stuff, but Illumina sequencing is much shorter, about sort of 75 to 300, up to 500 bases, which is still an awful lot longer than 22, but it is quite short. and then Sanger sequencing, anything up to 1500 is the limit. Yeah, so it really depends, but yeah, that is something that we need to think about. I think my oldest son got it, he's 8, and I explained it to him in those kind of terms, and he understood. I guess when you're doing a workshop, it's very one-to-one, and you can have conversations with people, and you can see if they're understanding, and you can go into further detail. So it's much easier when you're face-to-face with people.
Yeah, I suppose it's where the Lego sort of facsimile breaks down, because if you actually built a piece of Lego that was 235 bricks long, that would be enormous, wouldn't it?
Yeah, it would snap quite easily, wouldn't it?
Yeah, it would be quite well. Yeah, you know, DNA can get sheared, so.
Yeah, and it would take a long time to get it through the sequencer. Actually, it goes quite quickly, but if you saw the video of it pulling through, if you had something that long, you'd be standing there for ages waiting for it to go through.
Yeah, you might lose the children's attention, mightn't you? Yeah,
I think so.
Okay, so I think we've run out of Brickopore specific questions, but thank you very much, Andy. And thanks for a special thanks to you, because you did have to suddenly adapt what it was that you were going to do for us. So thanks a lot for sticking with us and
that's okay.
And giving it a go. You've been very game. So thank you, Andy.